So a little background of how I'm using plant soil feedbacks in this. Some contention, I use it to mean heterospecific feedback, soil mediated effects. So it could be microbial, it could be chemical. But just to kind of illustrate what I'm talking about, imagine a plant, hypothetical plant, it's growing at a certain size. This pot represents a volume of soil. So this, of course, could be in the field. Now imagine it goes away, harvested or whatever. Then another plant comes into this place. It could be the same species. Here I'm using a different species. That grows for a while. Then you have the same plant coming and growing in that original soil, the original plant. In a positive feedback, it grows bigger. And this could be caused through resource deposition, say nutrients are increased in the soil from that intermediate plant, mutualist accumulation, so bacteria, fungi that are growth promoting for this original plant. So it grows better after the growth of the intermediate plant. Alternatively, you can have it growing small afterwards, and that's a negative feedback. So that could be through resource depletion. It takes out nutrients, mutualist depletion. There's less of these growth promoting taxa, often microbes. Could be buildup of pathogens, again, also microbes, usually microbes. And then allelopathy is chemicals produced by this intermediate plant that get in the soil and can suppress the growth. So that's how I'm using it. My system, so negative feedbacks can play a role in invasions, and that's what we were following up with this study. We were working with cheatgrass. So cheatgrass invasion, cheatgrass came into the West in the 1880s, and then over the course of the next half a century came to really dominate a lot of areas in the West, especially what used to be sagebrush areas, dominated areas. So you can see various states of sagebrush losing to cheatgrass. How does it do this? Well, so it comes in after a disturbance, often a fire, and then it will dominate afterwards. How does it maintain dominance? Well, it germinates really early in the spring, whereas most natives germinate in summer. So it can kind of get a competitive edge by germinating early. Also, after it comes in, you can see it forms these dense mats. So those will burn really quickly and easily and frequently. So that will then, like the fires, will prevent reestablishment of natives. But also, it's been suggested, and there's some evidence that they're altering the soil through negative feedbacks. So this is what I wanted to look at. My research question, is cheatgrass exerting negative feedback on sagebrush? And then how does that compare to feedbacks of a native grass or to conspecifics? So what kind of feedbacks do sagebrush have on themselves? And then I wanted to look at some mechanisms, like follow up some mechanisms to explain that. Nutrient depletion was one that we looked at, and reduction in mutualists, and the increase in pathogens. So we were looking at fungi and bacteria. Experimental design. First I had a conditioning phase. So each of the different colors of pots are the different treatments. Grew sagebrush, squirrel tail, which is a native bunch grass that co-occurs with sagebrush, and then cheatgrass, the bad guy, and the controls were open, just left open for that 6-week conditioning phase. And I planted them at different densities to see if there was an effect of, well, more conditioning is more bad. So after 6 weeks, harvested, weighed the conditioning plants, then put them into the test phase of 9 weeks. So the 6 weeks was because cheatgrass starts flowering after 6 weeks, and I didn't want to deal with reproductive stuff. And then say 9 weeks because sagebrush is pretty slow growing. So again, the 4 treatments, 1 sagebrush seedling in each pot. Then harvested and I measured root and shoot biomass of the test sagebrush, tissue, nitrogen, and phosphorus content, colonization by fungal symbionts. So we were looking at arbuscular mycorrhizae, which sagebrush benefit from having, and dark septate endophytes, which probably are mutualists. It's not totally clear, but they seem to have some plant growth or growth promoting effect. And then we did amplicon sequencing to look at the fungal community and the bacterial community. I just got those data, so they're pretty raw yet, but I didn't add that in. And so again, feedback here on defining as the plant conditioned values minus the control value, the open value. So it's like, did it have a negative effect, or did it have a positive effect? What did I find? Negative feedbacks on test sagebrush. So here, the 0 line is the mean of the control treatment. So you can see everything was negative. But sagebrush is less negative, so it feeds back less negatively on itself than did squirreltail, the native, and cheatgrass. So just to give a little context, negative 0.4 grams, this was like huge. These were very tiny. A lot of them were just barely growing, or they didn't even get past just when I transplanted them, like a little bit larger than germination. So it was a huge suppressive effect. The letters are the significant differences between groups. Sorry about pink. It was like the pink was the pot that I found, and so I wanted it to match the pot colors. And then people pointed out how ugly that was, but I didn't. Anyway, so negative effect. First thing I looked at was, well, is it nutrient limitation? This is a hypothetical graph of what you would expect if nutrient limitation was occurring, where the control 0, and these are this is tissue nutrient content. So nitrogen or phosphorus, you would expect that if nutrient limitation was occurring, that they would be getting less nitrogen or phosphorus into their tissues after conditioning. But that you wouldn't see that in sagebrush, or not as much, because it didn't have as much of an effect, negative effect. What did I see? Not that. Control 0, sagebrush actually had some limitation, less nitrogen got into the tissues after conditioning with sagebrush. But cheatgrass actually increased nitrogen content in the tissues. It's not actually that surprising. Cheatgrass has been found to increase nitrogen in the soil. But either way, we don't see the same pattern that we see with the feedback, suggesting that it's not a nutrient limitation story. Squirreltail is intermediate. And then phosphorus just was a boring graph, so I didn't put it up. No differences. Now we looked at mutualism depletion and struck out a bunch. Arbusco mycorrhizal colonization, no significant differences between groups. Dark septate endophyte, no differences. And then looking at fungal communities, so this is using genetic sequencing to describe communities. Fungal communities did not vary. Dark septate endophytes didn't vary. Just random, didn't see any patterns. Unfortunately, the Arbusco mycorrhizal analysis failed, and then we ran out of money. And then fungal species diversity, so Shannon diversity, alpha diversity, didn't vary between groups. Bacterial communities, just got this back, don't know here, this is just the genus of squirreltail. And the sagebrush, didn't get it. But there's an interesting pattern where cheatgrass has more similarity between samples. So cheatgrass is kind of more similar to itself in terms of the bacterial communities than were the red, which is open, and the squirreltail. But again, it doesn't, I don't know what to do about this. But it doesn't fit the pattern that we saw before, So now we have a difference between cheatgrass and LMS. I'm still working on that. I don't know what this is. And also species diversity is higher in cheatgrass condition treatments. So, despite the really negative feedback that it had. So my conclusions here are invasive and the native grass both have strong negative feedback on sagebrush. The mechanisms I looked at, nutrient depletion, of course it's just nitrogen and phosphorus, but those are really important nutrients. And I didn't find anything. Could be other nutrients, certainly. Micronutrients or potassium. I did find strong nitrogen limitation based on nitrogen phosphorus ratios in all of the soil, all the conditioning treatments. And then mutualist depletion, I don't really know, but I certainly didn't see anything that fit the pattern, a pattern that would explain the feedback that I saw. Arbuscular mycorrhizal community composition, I don't know. There wasn't any variation in colonization, so they had lots of mycorrhizae in all of the treatments, but it could be that they're less beneficial species that they're getting. And then I have to acknowledge that, especially with bacteria, a lot of these identifications, you're using short sequences, so a lot of times I wouldn't be able to identify it past order. So it's possible that there's shifts happening in there that I'm not picking up on. A lot of these, like the plant growth promoting taxa are, in the same family, even genus, as a pathogen. So there could be shifts that I'm just not picking up on. And then allelopathy, I didn't look at it. But interestingly, what I found one of the kind of most puzzling results is that I had huge variation in my, what I call my conditioning strength. So the size of the root mass, the size of the conditioning plant. Here, you just see that cheatgrass can get these giant roots. A lot of times they would be almost root bound after 6 weeks in my pots. And then other times there would just be a little bit of roots. And I saw no pattern in the, no relationship with the strength of the feedback. So, in other words, even a tiny little bit of cheatgrass would have just as much of a suppressive effect as like these huge root masses, which is weird. And it's not density dependent. So usually with allelopathy, there's some kind of dose dependency where more of it is worse. I didn't see that. And you would also maybe expect that with mutualist depletion. So puzzling, didn't really answer. We found this interesting pattern.